Okay, so let's start looking at the parts of DNA. Now, DNA is a polymer, and if you recall back, we were talking about polymers several times. We said a polymer is a string of molecules, and these little molecules that they're a string of are our monomers, and in the case of DNA, it is a nucleotide. So here we have a picture of what our nucleotide is. If you notice, there's a phosphate group here. We have a sugar, and the sugar is going to help us determine which type of nucleic acid that we have, whether it's DNA or RNA. And then we have our nitrogenous base. And our nitrogenous base is what's going to code for the proteins, and we'll learn about that a little bit later on in the unit. So when we take our nucleotides and we join them together, you'll notice that we have one of them is here, and then another one is down here, and then another one here, and you can see how it's this string together effect of a polymer. And they're joined here with the sugars and the phosphates. The sugar of one nucleotide will join with the phosphate of another nucleotide, and this is our bonding here that makes this polymer grow. So as I hinted, we have two different types of nucleic acids. We have our DNA and our RNA. Our DNA, they both have the phosphate groups. Our DNA gets its D from deoxyribose, and our RNA gets its R from this ribose sugar here. So our sugar determines what it is. And then when we look at our nitrogenous base pairs, they're all going to contain adenine, cytosine, and guanine, but our DNA will have thymine, and our RNA switches it out into uracil. So that's just a real quick overview of the parts of DNA. Remember, DNA is the polymer. Okay, RNA is also going to be a polymer, and both of those are going to be made up of these units here we call nucleotides. Okay, let's meet the nucleotides here. Notice that we have two different fundamental structures. We have our two-ring nucleotides up on the top here, and then we have our single ring down here. Now, if you notice, there are 5 nucleotides. We are going to have some that are going to be in DNA. We'll have some that are going to be in an RNA, and the bulk of them, 3 of them, are actually going to be in both. So let's take a look at DNA first. DNA is going to have the guanine and the cytosine, as does RNA. RNA is going to have guanine and cytosine. Both DNA and RNA are going to have adenine. But here's where we see the difference. With DNA, we use this base pair thymine, but when we get to RNA, that thymine is switched out to uracil. And that's one way we can tell if we have DNA or RNA is by looking at the bases. And if we have the uracil, it tells us we have RNA. And if we have this thymine, then we know we have DNA. Now, the discovery of DNA structure is kind of an interesting tale. It talks about 5 different scientists that we want to mention real quickly. One of the first was Erwin Chargaff, and we'll talk about him real quickly. You can see a picture of him over here. What Erwin Chargaff realized was that when he took DNA and he broke it down and measured its components, he realized that the amount of adenine was always equal to the amount of thymine. He also found that the amount of guanine is also equal to the amount of cytosine. And when we look at the structure, we'll kind of see how that starts to work out. Now, we also get to meet these 2 individuals here, Wilkins and Franklin. Franklin probably is the least known of all of these, but lately she's gotten the press that she's been due. What these 2 individuals did is they took a picture, an X-ray picture of DNA, and saw that its structure. And by looking at that picture, they inferred that it had to be some kind of a spherical or helical structure. So they had that information. At the same time, Watson and Crick, who got most of the fame for determining the structure of DNA, they were building models and trying to make this work out. And what they noticed is when they saw this picture here, they realized what it was, that it would be a double helix. So there were 2 strands in DNA. So DNA has 2 strands, and those 2 strands are going to be twisted on each other. Okay, and then what they also started to look at is they started to look at, from the x-rays, how wide these 2 strands had to be, and they came to the realization that if they took a purine, which is a double ring nucleotide, okay, so you can see 1 ring and 2 ring over here, and a pyrimidine, which is a single ring, if they took 2 of these purines together, it was too wide. If they had 2 of these single rings, it was too narrow, and the only way it made it fit the data is if they had 1 of each. So if we go back and we look, we notice that we had our 2 ring structures and our 1 ring structure, And those ones here, each of them had a complementary base pair, and we can start to see from Chargraph's work what these base pairs are going to start being here. So finally, we got to what we know DNA looks like. We have our complementary base pairs in DNA where we have adenine and thymine, and we have guanine and cytosine. So when we notice, and we have our two strands here, you see our strand 1 and strand 2 there, what we notice is these ones will always bond to each other. So we always have the adenine thymine bond, and we always have the guanine cytosine bond. Now, if you recall back to our earlier slides, we saw this sugar phosphate backbone, and that builds up this edge, this rail, so to speak. And we'll notice that in both strands, and those strands are going to twist behind. And a lot of times it's like a spiral staircase. So the rails of the staircase are going to be this sugar phosphate. And then we'll notice that the steps themselves are going to be these actual bonds of the base pairs. And that's what our DNA structure looks like. You can kind of see it over here as well. It's a double helix. And the lessons will go into it a little bit more for you. Good luck on the lessons. And as always, we'll see you in the next video. Okay, so when we talk about the discovery of DNA replication, what we're talking about is the Hershey-Chase experiment. And what Hershey and Chase did is they found these bacteria cells and they infected them with a virus called a bacteriophage. And these viruses would then create a change in the cell or in the process of the bacteria. So we knew that there was some genetic information being transferred. At the time, there were two different groups that thought what it could be. Some thought it was a protein that coded all the information, and the others thought it was these nucleic acids. So what Hershey and Chase did is they set up two different experiments. They had one up here and the other one is down here. And what they did is they used a radioactive tracer. So in the first experiment, they took this radioactive tracer and they attached it to the protein coat of a virus. So they thought that if the proteins in a virus were what had this information, molecules that were being passed on, that by doing so, when this bacteriophage was released on a cell and then transferred the genetic material, if it was a protein, then there would be protein radioactivity. But unfortunately, there was no radioactivity in the cell this way. So what it led was the idea is that it was not a protein. Now they had to check to make sure that it was the nucleic acid, in fact. So they ran the second experiment here. And in the second experiment, they put a radioactive tracer in the nucleic acids. And when they released the phages onto the bacteria again, what they noticed afterwards is that radioactivity was actually inside. And what they found out was that it was this nucleic acid, and in fact it was DNA that was where the information was being stored, and then later on this DNA would then be replicated through the cells, and they could track these tracers through future generations and see how it passed on. Now, one of the terms that you're going to hear in this unit is semi-conservative replication. What this means is that, quite simply, when we make DNA, when we have to make our two strands of DNA in interphase during the S phase, right before we have mitosis, what we're going to notice is that this DNA, remember it was a double helix made of two strands, is going to break down the middle, and it's going to use each of these strands as a template. So because those base pairs only bond with a certain one, they have that complementary base pair property, it's going to be serving as a template. Will make sure that when it is recreated or when they do duplicate or replicate the DNA, by using one of the strands as its template, it's going to force the other strand to be lined up properly. And that's what we talk about as semi-conservative replication. So how does DNA replicate? Well, we found that there are these origins of replication, and these are sites on the DNA strand where it will seem to unzip, like we can see here, and form what we call these replication bubbles. And within these replication bubbles is where we see this DNA being copied, and the net result is going to be these two DNA molecules. Now, if you remember, we were talking about that conservative replication. Well, here's an original strand. Notice we have that same original strand now serving as a template for a new strand, and then we end up with our two DNA molecules. One is going to be the original strand, and one will be this new strand that was just formed. Okay, so what actually happens in these replication bubbles? What are we seeing here? Remember, we have our replication fork, which is where it's splitting apart, okay, where it goes from being a double strand here to this open DNA replication bubble here, and where formation of our new strands occurred there. Now, there's an enzyme involved, it's called DNA polymerase, and what DNA polymerase does is it goes along the old strand of DNA, the original, the template, and it will add these complementary base pairs to it, and that builds this second strand, this new strand of DNA. Now, we have two different ways that it works. In the upper strand here, what we notice is the DNA polymerase, it's going this way. Okay, it'll open up here and it's just going to make one long molecule as it travels along this way. This is what they call the leading strand. Down below, the direction of replication is going to be a little bit backwards. And what it does is it actually makes small little segments, and then they put those small little segments together. And this is what they call the lagging strand. So the leading strand is going to go and it's going to make one smooth process, whereas the lagging strand is by chunks, and they'll kind of chunk it together like that. So let's take a little closer view as we go through this process. And to do this closer view, we'll start off with our origin of replication. And remember, our origin of replication was quite simply just a portion of the DNA that says, hey, start making copies here. So in our case, it's going to be right here. Okay, so here is our final product. We now have two new DNA molecules. Remember, each of the molecules is going to have its original strand, which it used as a template, and that's what gives it its semi-conservative replication. We'll also have a new strand here, and the new strand is lined up from the DNA polymerase, so there's very few mistakes going through, which is what we want it to do. And we'll notice that both DNA molecules look that way. They both have an original strand, and then they have a new strand attached to it. So that pretty much sums up our video and what we talked about today with DNA replication. As always, the lessons will go in a little more detail. Good luck with the lessons, and we'll see you in the next video. Okay, let us begin by talking about transcription. Transcription is the process of transcribing or changing our DNA, which is the blueprint, into something usable. And that usable thing is going to be RNA. And in fact, we're going to make messenger RNA, and that messenger RNA is going to be able to travel outside of the nucleus into the cytoplasm, where it'll meet up with some ribosomes, and that's when we can make the actual proteins. So this process is going to go how we get from DNA to RNA. Okay, so to begin our process, we have to start with the DNA. Remember, the DNA is in the nucleus. It's a double-stranded molecule, and this is our genetic material. This is what is the blueprint to how we build our cells. So this has the codes for all of the different proteins that we'll need to make our cells. And we need to change this DNA to RNA so that we can take it out of the nucleus and start manufacturing those proteins. Much like replication, you'll see that we're forming a little bit of a bubble here. However, we are going to do it a little bit differently. Here we have a promoter, which is going to separate the two strands of DNA, and then we have a terminator, which will end this separation. So instead of separating it and making a whole complete strand, we don't need that. When we're talking about making the RNA to make a protein, we're going to have a section of the DNA, a chromosomal gene per se, and that section of DNA is what we're going to copy off of. We don't need the entire thing. Okay, much like the replication, in transcription, we'll see that inside this bubble, we're going to have this molecule, RNA polymerase, is going to go along the DNA molecule. So this is our DNA molecule, and it's going to serve as a template to make the RNA. So now as we're going through, we'll match up our base pairs. Okay, here we have a guanine matches to a cytosine, and a cytosine to a guanine, and a thymine to an adenine. But notice here we have our adenine, and now it matches up with uracil instead of thymine. So this uracil is what makes it that RNA, okay? And we're forming a single strand of mRNA. And remember, RNA is going to be a single strand, DNA is a double strand. After we've created the mRNA, the DNA will close back up, and it'll go back to doing what it's normally doing, probably going to open up again and make more messenger RNA. Might be the same gene, might be a different gene, but that's how the process goes, and we're left with this mRNA molecule. And remember, the mRNA, unlike DNA, is a single strand, and it also has this uracil instead of the thymine that we saw earlier in DNA. Okay, so now that we have the messenger RNA, which is the template for making a protein, we have to make the protein. And that process is called translation. Now, translation happens in the ribosomes, and the ribosomes are made up of ribosomal RNA and some proteins. And we'll see what these do in a little bit. We also have this transfer RNA molecule, and the transfer RNA molecule has an amino acid attached to it. And when we string these amino acids together, we form a protein. Now, the mRNA is going to have its bases broken down into these groups of 3 that they call a codon. If you notice with the transfer RNA, we also have a group of 3 here. So this codon tells us which amino acid we need to make the protein, and the transfer RNA will bring the proper amino acid. And the way that it brings the proper one is only its anticodon, or its 3 base pairs, will line up and make a match. Okay, so let's go through the steps of translation. We're going to start off in the nucleus, and remember, inside the nucleus is where we can find our DNA. Now, our DNA has gone through the process of transcription, and it has made this messenger RNA, and this messenger RNA is able to exit the nucleus through these pores, and that's what gives us the material out into the cytoplasm so that we can start building proteins. Now, all of our proteins will start with a start codon. And remember, the codon is our 3 bases. Okay, it's a little system like that. So those 3 bases will come together. The start codon for all of the proteins is going to be this one right here, this adenine, uracil, guanine. Okay, so that will come and that will line up into the ribosome, and the ribosome will take the mRNA and feed it through in this direction. So it first starts off with this start codon, and it says, hey, we're gonna start making proteins, and that's what the start codon does. Now, if you remember, we have our start codon, which was these 3 base pairs here. We then bring in our tRNA molecule. Now, our tRNA molecule has the anticodon, which is going to be the 3 complementary base pairs for the codon. And you'll see that the adenine and uracil bond up, and the cytosine and guanine bond up. And this one here, because it only matches up this way, it brings in one specific amino acid. And that amino acid begins to be the first of many in our protein polymer. Okay, so here we see the end of the process. So we missed this middle part. Remember, the mRNA has traveled through the ribosome this way, and we have this broken down into our 3 little codon systems here. Okay, and what these 3 little codons do is, remember, they line up with an anticodon on a tRNA molecule, and they bring in a specific protein. So we can see how these specific proteins are all lining up together to form this protein polymer. When it gets to the stop codon, and there's several different stop codons, and this one, it's this UGA, what that's going to do is that will tell the ribosome to stop bringing in more. That's the end of it. So even if this chain went a little bit longer, what it's going to do is here, when we attach this one, it cleaves it off, and that ends our protein for us. So now we have our completed polypeptide, our protein. The amino acid chain has been released, and it becomes this polypeptide. And then, because of the hydrogen bonds between these different amino acids, it'll start to fold and take shape and become the protein structure that's necessary for the cell to do what it needs to be doing. Now, sometimes mistakes happen and we have a change in the DNA sequence. And that can happen in the DNA, or that can happen during DNA replication, where we can make a mistake, or it can happen in the transcription when we're making the mRNA molecule. And what we notice is we have our DNA here, and it kind of shows us what it is. Now, my DNA is going to match up to my mRNA, which then matches up to my tRNA, and then we have our amino acids here. So we can see how this process is going to make this protein that'll look this way. Now, the easiest type of mutation, or the one that we see quite often, is this substitution, and that's where you just make a mistake. So here in our example, if we look at it, we have our first codon is still the same, our second codon is still the same, but in the third codon, what happened was we took this C out and we replaced it with a T. So the cytosine went out and the thymine came in, so we have a little bit of a difference here. Now, because it's different, it's going to bond to something different here, this A, and we notice that here in the original protein, we had a CGU, which formed arginine, and here we formed histine. Okay, so we can see that we have this little bit of a difference, and that's going to form a difference in the protein. Sometimes it's noticeable, sometimes it's not, so it's going to vary, but our first one is where we're just substituting one for another. Now, there's two other kinds of mutations that happen every once in a while. One is a deletion, where we'll actually just be going along and we'll actually just forget this one. Okay, so it's just deleted out of there. And then its opposite is going to be this insertion, where we'll just add in an extra one also. And these are our major types of mutations that we see. And you can see how they're all just going to be mistakes. So even in the conservative, it's semi-conservative replication and semi-conservative transcription, it's because we're using one as a template. Every once in a while, we'll still make mistakes. Okay, so the lessons will go over these in a little more detail. As always, good luck with those, and we'll see you in the next video. Okay, so viruses are made up of two things. They have two different parts. The first is this protein capsid here, and this is going to be the outer shell. So it's made up of a head here, it has the tail sheath here, and then the little tail fibers here, and those are all proteins. The other structural part of a virus is the DNA that's found inside of the head here. Okay, so we have the protein capsid, and then we have the DNA material, and that's all that's in a virus. Okay, so how do viruses infect other cells? Well, as you can see, our little virus friend over here has attached to the outer membrane of our cell. In this case, it's probably going to be a bacteria. And then what it will do is it attaches in, and then it will inject in its DNA. And you can see its DNA materials being done here. So the DNA that was up in here is going to be injected into the new cell. So here we can see that our virus is already injected in its DNA into the host cell. Now it's called the host cell because now it's hosting this new genetic material. It is at this point that the virus, the DNA in the virus, has two pathways that it can follow. One is the Lytic cycle and the other is the Lysogenic cycle. So let's take a look at the Lytic cycle first. So in the Lytic cycle, what happens is the host cell will start to make copies of the viral DNA. Okay, and it'll start making the necessary proteins. So it'll undergo replication and make copies of the viral DNA, and then it'll go through transcription and translation and start making these proteins. Okay, so we'll start seeing that what we're doing is we're making more and more viral DNA. Now the proteins will fold over and they'll form more of these capsids for the virus, and the DNA will then come in there, and what we'll notice is that we're starting to assemble many viruses. So the cell that was injected with this DNA from the virus has then started to make more and more viruses. And the net result is this host cell will then rupture and release more viruses out, which can then go on and repeat this process. So in the Lytic cycle, what happens is the DNA comes in, and then that DNA is used right away, and it's used to make more viruses, and that's how viruses can spread, either in an organism, or in the case of this here, we have one single-celled organism that's just manufacturing more. It becomes a virus factory per se. Now the Lysogenic cycle is a little bit different. What happens is we still have the injection of the viral DNA, but here what you'll see is it's kind of bonded up and it's hiding inside of the host chromosomes. So in our bacteria that has one chromosome, what we'll see is this viral DNA just kind of hides. It hangs out. It's not doing anything. It's just there. Now, because it's become part of the DNA, as that bacteria cell goes through binary fission and it makes more bacteria cells, what we're going to see is this viral DNA is going to be copied, it's going to be replicated, and we have all of these cells now are being infected by the viral DNA through a normal cell process. Now, we can have some kind of a stress event for the cell, or it can be a time event. Normally, it's some kind of a stressor. When that happens, this viral DNA is going to exit the chromosome. So it used to be attached in here, and it's going to separate itself out. Once it separates itself out, then it will convince the host cell to enter the Lytic cycle. So once it's out here and it's on its own, it'll start to replicate itself, it'll start to make its proteins, it'll make more of these little viruses, and then the cell burst and release all these viruses out. So the lysogenic cycle is more of just a wait until the right time, and then once that happens, then all of these cells will go through the whole process. So here we can see our real quick overview of these. We have our lytic and our lysogenic cycle. Remember, it all starts when this virus comes down and injects its DNA into its host cell. And from there it determines. It can go into the lytic cycle where it's going to manufacture more viruses and release them, or it can go into this lie and wait cycle, the lysogenic cycle. The lysogenic cycle will go until it hits some kind of a stress, and then once that happens, it kicks in the lytic cycle. And that's how viruses reproduce. So viruses aren't living per se because they can't reproduce on their own. They have to use these other cells for this to happen. Okay, so that's it for our video. As always, good luck in the lessons, and we'll see you in the next video. So what is biotechnology? Quite simply, biotechnology is the use of genetic information to make our life better. And we can kind of see how it goes here. We can go through a simple example of it. We have our bacteria here, and if you notice, we're going to integrate in some new genetic material. When we do that, this is in the form of a plasmid. This little ring of DNA is called a plasmid. That's going to integrate itself with the chromosome of the bacteria, and then as the cell replicates, it's going to pass on that information. And whatever we decide to put on this plasmid will allow the cell to do the same thing. Okay, so let's take a look at some improvements in medicine and food. First off, in medicine, let's take a look at insulin production and the treatment of diabetes. We used to collect insulin from animals that were able to produce it, but now by using that plasmid technology, we were able to introduce it to a bacteria, and then we can have that bacteria produce this insulin that we can then use for treatments. The other big one that we see a lot of is this idea of food and the GMO, the genetically modified organism, when it relates to food. However, genetic modification really started 10,000 years ago. That's when we started to domesticate plants and animals, and through selective breeding we could choose what we were trying to get, what we wanted, what was the end product, and that's what we bred for. In the 1700s, we started crossbreeding with plants, and within just a few species, and we could kind of see that crossbreeding in Mendel's work and how that was working out. In the 40s and 50s, we started to see about introducing genetic variation into the gene pool of plants. So by this point in time in history, we had a fundamental working idea of genetics and heredity, and we were starting to use that information to start making plants a little bit different. In the 1980s, we had more controllable methods of genetic engineering, so we were able to start introducing just that genetic material that we wanted to and modify the plants that way. And by the 1990s, we were starting to have our first GMOs in the marketplace. So there's a lot of misunderstanding and misinformation about GMOs. We've been doing it for 10,000 years. So it's up to you. You can look at it as you will. The lessons will go through it a little bit more for you, and you can always research it a little bit more on your own. Okay, so let's end by looking at a few medical advances. By knowing the genetic material, what we're able to do is we're able to see how these genetics can play into several diseases. One of the ones we found out was with Down syndrome, that we had this non-disjunction during mitosis. So here, as we would have expected the chromosomes to split this way, through non-disjunction we end up with one that comes this way. And what that means is that we're going to have an extra chromosome, and that's going to be the cause of Down syndrome. We also have advanced in the field of cancer research, and we found out that a lot of cancer is genetic in nature and how we respond, or how our cells are going to respond to this. So we do have a genetic predisposition to or against having cancer, but also we have the idea of mutations, and we talked about some of those. And some of these mutations can cause cancer cells to form and grow, and that's where the cancer comes from, is a mutation in a simple replication error. So that's it for our video on biotechnology. As always, enjoy the lessons, and I hope you do well on them, and I will see you in the next video.